Could I have the first slide, please? Well, it's a pleasure to have Maxine Linial visiting us here at Cold Spring Harbor again to give a keynote lecture. Maxine was an undergraduate at Cornell University. Well, I should start with where she went to high school. She went to high school in Brooklyn, and she retains a little bit of that New York accent, even though she's lived on the West Coast for quite a while. And notably, she was in the same high school where I believe Bobby Fischer was a year or two senior to her at the time. Barbra Streisand too. Awesome. She later went to Cornell as an undergraduate, where she was fascinated at that time by biology already. And another trivia bit was at Cornell, she was friends with the future Mrs. John Kaufman, Marty Surek, I believe. And so she had known Marty, I think, before John knew Marty, and would later keep up with them, of course. After finishing her undergraduate at Cornell, Maxine went on to Tufts University, where she did her graduate work on phage with Mike Malamy. And the department still remembers her fondly, as I will show in slides to come. After her graduate work, Maxine later worked in the lab of Peter Vogt. Peter was initially in Seattle at the University of Washington, but moved during the time she was with him to Los Angeles, where he established his lab again at USC. And it has, of course, had been and a powerhouse with regards to retroviral researchers that passed through there. After working in the Vogt Lab, Maxine found her way back to Seattle again, where in due time she received an appointment as an investigator at the Fred Hutchinson, a member eventually at the Fred Hutchinson, where she did seminal work in retrovirology. She was the one of the first to map RNA and protein, RNA domains in viral RNA and protein domains that interacted with the viral RNA for avian retroviruses, isolating the first retroviral packaging cell line, I believe it's SC21Q1B, that gave notion to the, provided hope towards the development of retroviral vectors and a field that, it has been enormously important in many ways beyond gene therapy and our basic sciences. She contributed to our RNA packaging studies for a while, worked in HIV, as we'll hear, and then later on did seminal work in foamy viruses, helping to usher in this field in the molecular era, in a sense. She has an amazing record of service. She is a fearless individual who has an enormous amount of integrity. As you, if you didn't know before, you will learn very quickly that she is extremely sharp. And what is not always appreciated until one gets to know her, and when one gets to know her, you're just amazed by it. She's a selfless individual. She's helped countless people at various stages of their career, junior PIs, graduate students. She often, without prompting, without asking, behind the scenes. She complains when people call her an inspiration, but 5 years ago, 10 years ago, 20 years ago, Maxine was an inspiration and a role model to many scientists, and she continues to be in that sense. She, in fact, told John Kaufman at one time to give me a job, for which I'm thankful to. I want to just, her friends are enormous in terms of their numbers and their devotion to her and their caring for her. And there was a tremendous response when they heard she was participating in the retrovirus meeting. And many people offered to share stories, as well as a few pictures of years past when Maxine was at her different places. And I'm just going to flip through these before I give you Maxine. So, a reminiscence for the audience. So this is Maxine when she was at Tufts University. She's right over here holding the glass of wine as a graduate student. I can't identify the other people in the shot. Here is a photo later on at Tufts University where Maxine's returned for a reunion. And this is Maxine, of course. That's John Kaufman a few years ago. That is her close friend, Naomi Rosenberg, who is a dean at Tufts and was once Alan Engelman's graduate advisor. And here's Mike Malamy, her graduate advisor. and the department chair. Yeah. And here is a picture that Robin Weiss shared of Maxine when she was in the Vogt Lab. I believe this is in USC, this picture. John Weick's in this picture, Bill Mason's in this picture. We can see Maxine here. And Robin shared that when Maxine left the lab, she was excited and set on exploring new areas beyond retroviruses and interested in expanding to different areas. But I think fortunately for us, retroviruses eventually drew her back. And here's a Vogt Lab reunion picture with Robin and Eric Hunter with Maxine. And here's Peter with Maxine. And Karen, her close friend, who is not a postdoc with Peter, but was a postdoc with Tony Hunter. And later, as I mentioned, Maxine moved to the Fred Hutchinson, where she was a PI. And I love this shot. This is a party, I believe, at Maxine's home. And in this picture, we see a notable and important papillomavirus researcher, Denise Galloway, Ginger Zakian, who's made tremendous contributions to telomere biology, Faith Yashimura, a transcriptional biologist, Meng-Chao Yao, a developmental biologist, Julie Overbaugh, a friend to many here, a prominent SIV, HIV researcher, and Mark Gradine, a transcription chromatin biologist. And there were many, many shots of Maxine and her friends that were provided. I'm just showing a couple. Here's a trip when she visited Robin in England with Karen. And here's a group that I saw many shots of, Susan Ross, Karen, and Naomi with Maxine. And I really love this picture. I don't know who took it. Maybe Suzanne had, or maybe Naomi had, of Suzanne, Karen, and Maxine looking backwards towards the photograph. In any case, we're going to, Maxine obviously continues to contribute to her fields of interest and fields of study. She's received a grant in the last year and a half. She's published an important paper on the role of an RNA interacting factor in the biology of foamiviruses, and you will hear these studies. I'm going to remain up here while she gives her presentation in case I can assist. She was on my graduate committee years ago, so I'm very used to taking instructions from her and good advice from her. All right.
Okay. So I was going to introduce myself. I just wanted to say that I've been at the Hutchinson Cancer Center since 1975, so that's 37 years. And I am still active there, even though a little over 2 years ago I was coming back from a run in a crosswalk with a green light when a car ran me over. And this led to life-threatening injuries and then brain-threatening injuries. And fortunately, I've recovered from most of them, except I'm totally blind. So this has really made it very difficult to do science, but I'm trying to prevail. I have the Hutch hires a scientific reader for me, so that makes things a little bit better. I also want to say that I had brain injuries, TBI, and some of this has led to some deficiency in memory. I mean, I'm getting old anyway, but this is this is aggravated. So I just want to apologize in advance if I cite the wrong people for some of the studies, or don't cite someone, or the dates are wrong. I find it very difficult to correct myself, because when you're blind, you can't just go to the internet and look things up. Okay, so let's see. On the
Easel.
Oh, the easels.
Yeah, over here. Two more steps.
Okay, all right.
There you go.
Okay, so I'm going to talk a little bit about my history in retrovirology and some of the influence Cold Spring Harbor has had on some of the research I've done. And at the end, I want to tell you about some stories about foamy-virus that are pretty recent. But I'd like to start off and tell you that my introduction to retroviruses was less than auspicious. And I think this story, if it wants to inspire anyone, maybe some of the younger graduate students, to make them realize all is not lost. I started at Tufts Medical School in the microbiology department as a graduate student, and I didn't realize till I went there that I was really interested in animal viruses. I did rotations and wound up working with the only animal virologist in the department at the time who worked on polyomavirus. And I was really excited. I was going to look for early proteins in polyomavirus replication. But he chose to move to the Southwest, and I chose to remain in Boston. So I had to find a new advisor, Mike Malamy, where I worked on bacteriophage. It turns out the department was very new. I didn't realize how new it was, but I was one of the first grad students, and the other two students who entered with me already had master's degrees. So when it came to the prelims, the preliminary thesis exam, you know, the general question exam, they decided to be nice to me, and they decided to put some animal virology questions in there, although there were no animal virologists in the department. So I sat down to take the written exam, and everything was really, really easy, and I answered all the questions until I got to the last question. Part A, describe how a DNA virus could cause a tumor. I went, wow, I've been in polyoma. I know all about polyoma and SV40. This is a piece of cake. And I just wrote and wrote. Part B was describe how an RNA virus could cause a tumor. I looked at that question. I said, what? They must be putting me on. This can't be possible. And I left it completely blank because I'd never heard of one. I passed anyway. That was the only thing I got wrong on the exam. So that was my introduction to RNA tumor viruses. Later on, when I was going to graduate, and this is around 1969, I wanted to work in a virology lab, and I had to leave Boston because it was one of the worst winters I could remember. And as I was digging my car out from its parking space in my landlord's driveway, I got to the point when I would dig the snow and try to get it on top of the pile next to me, it would just come back down on my head. I said, I've got to go to the West Coast or Europe. And so one of the faculty members went over to MIT, where there was a young assistant professor who turned out to be David Baltimore, who drew up a list of people that I could work with. And when I wrote the letters, the most wonderful answer was from Peter Vogt, who I went to do a postdoc with. And I wrote a fellowship, and it was to use an immunological assay to try to look for an RNA DNA intermediate in retroviral replication. And this was just I got funded, but then Temin in Baltimore came out with reverse transcriptase. So I never did this project. I went to Peter Vogt's lab, and could you just hold that? And he was just the most incredible mentor in terms of teaching us to use viral genetics to really understand fundamental things about viruses and also the cells they infect. And it was a great lab. There were all the other postdocs had experience in animal virology. It was a very hard learning environment, but there was Robin Weiss and John Wyke and Bill Mason, and later, as I was leaving, Eric Hunter came, among others. It was just a great environment. So, okay?
Right over.
And I could actually, as you'll see, if I get through this, that I could call this talk "What Goes Around Comes Around." But my first paper ever was in 1973. When I worked with Bill Mason, we looked at early mutants of RSV, and they were temperature-sensitive mutants, and we showed that they had thermal label reverse transcriptase. And I think this was the first and clearest indication that this was a virally encoded enzyme that was required for replication. So that was really exciting. And then as Vineet said, I drifted around for a bit before I met Paul Neiman back in Seattle, who was working on RSV and the Sark gene, and he offered me space in his lab and eventually gave me an appointment. He was one of the founders of the Hutchinson Cancer Center, and in 1975 I joined the Cancer Center and have been working on retroviruses ever since. So when I set up my own lab, again, whoops.
Sure.
Okay.
And there's a light.
Okay. And so I hope I don't write over anything else. My first important paper on my own was in 1978 when I isolated and described a non-conditional mutant of RSV called 21Q1B, a name to be forgotten completely, but it's a great password. And in 78, I published on this mutant, and then in 79 and 80, worked with Pete Shank, who was then, I think, in the Bishop Varmus lab, and we showed that this had a small deletion at the 5' end, and it was a packaging mutant that didn't package the genome. If I have one regret in my scientific career, I was not assertive enough about this mutant. And some people who I shall not name ran with it, and they might have quoted my paper once, but then took credit for discovering packaging in another retrovirus. So I never felt that I got sufficient credit for packaging. Okay, so I should just hold on to it. And I worked on this mutant for a long time. It had a quality of packaging cellular messenger RNAs, and when you infected something else with this virus, something I called retrofection, I showed that the cellular messages that had no viral sequences on them at all could be reverse transcribed and integrated in the new cell. And this was something that occupied my interest for a long time. Because I had worked on SC21Q1B for the next couple of decades, I was really interested in RNA packaging. And by then, Psi had been pretty well described, and a lot of people were interested in the GAG sequences that were involved in packaging. And I worked on this in RSV. A lot of people did in RSV and MLV, and then later HIV. And I will get back to that in a little bit. Okay, so Cold Spring Harbor has been very important in my career all these years. I think I've probably been to about, in the 40 years, maybe 35, maybe 33 RNA tumor virus meetings. I got to run it in 1990 with Paul Jolicoeur. And the exciting thing about that meeting was it was the 20th anniversary of reverse transcriptase. And I personally got to design the decorations for a large sheet cake with the pathway of reverse transcription. And Timon and Baltimore both attended and, you know, cut the cake and everything. It was great. I've seen a lot of improvements in the Cold Spring Harbor meetings and the retrovirus meeting over the years. Three that I just want to briefly mention. It was one when oncogenes left the meeting. Even though I worked on an oncogene through the 80s. The other was building this hall and replacing the one that was tiny. You had to get there really early just to get in, let alone get a seat. It was just a disaster. With that pointer that was about 20 feet long, and if your hand shook a little bit, it looked like you had complete palsy. And then the other thing that I really have noticed is that in the early days of this meeting and Cold Spring Harbor meetings, there was almost no women who even got to speak. All the women were given posters, and I'm not kidding, it's true. And now it's, you know, it's just good science. Nobody is looking at what your sex is when they invite you to speak or not. Okay, so those are three things about Cold Spring Harbor. In the early 1980s, I was working on packaging, and I had started working on MIC viruses of avian MIC viruses and the acute transforming viruses, MC29 and MH2. And then somewhere very early in the 80s, I came to this meeting, and Bill Hayward, Susan Astron, and Harriet Robinson reported the incredibly exciting result about promoter insertion and activation of MIC in avian leukosis virus-induced tumors. And this was so exciting. And then I spent quite a number of years in my lab too looking at ALV insertional mutagenesis, promoter insertion in the quail system that I worked on. And in 1989, I isolated the only new virus I've ever isolated, which was a retrovirus in a quail injected with ALV. It was a new MIC recombinant called FH3 that we worked on for a while. So we worked on MIC and we worked on packaging, mostly looking at the sequences in GAG that were involved with recognition of psi. And it was, here's NC at the end. We all know it's important that it's a full-length GAG, and I don't know if I can draw this, but I'm putting two little boxes. These are cysteine-histidine boxes, and mostly everyone thought that these were involved in RNA packaging. And one day in the late 80s, I can't find the date, maybe the early 90s, I came to this meeting, and Rolf Flugel from Germany reported the cloning and sequencing of the first foamy virus, then called human foamy virus because it was isolated from a new human nasopharyngeal carcinoma cell line. And if it had turned out to be involved in the cancer and it had been a human virus, it would have been one of the first human tumor viruses. But it had nothing to do with the carcinoma, and it turned out to be a chimpanzee virus, as shown later, that was zoonotically transmitted to the human from who the tumor had been cultured from Kenya. Anyway, when I was at this meeting, stay there, and Flugel described the foamy virus sequence. I looked at GAG. There were no cis-his boxes. I said, my God, it's going to package its genome in a different way. This is so exciting. I've got to work on this before anyone else jumps on it. So I came back and I got the clone from him of HFV, which is now known as prototype foamy virus, or PFV. And it turns out Foamivirus has been a wonderful system, but I got into it for the wrong reason, because it turns out the cis-his boxes, which are clearly important for packaging, are important for packaging, as a number of labs have shown, because of the basic residues in the cis-his boxes, the arginines and lysines. And the cis-his is just kind of a scaffold for the structure of the GAG. And when you look at Foamivirus GAG, at the 3' end, or the C terminus, there are 3 GR boxes, glycine arginine-rich boxes, and it's actually the arginines there that are important in packaging. So even though superficially the sequences look very different, probably packaging is by a very similar mechanism of the C terminus of the GAG protein interacting with a specific RNA sequence. So I got into the... Okay, so I got into Foamiviruses for the wrong reason, but I think they have just been fascinating. So my lab still worked on MIC viruses, still worked on packaging of in RSV and also HIV, but starting in the 90s, there was a slow transition to Foamiviruses. And I want to tell you some of the major things we discovered. And let's see what time. I'm sorry, I'm going to keep punching my little watch, which talks to me.
The time is 7:57.
So I know how much time I have left. I can't look at the beautiful clock or anything. Okay, so just as an introduction to Foamiviruses, these are viruses that are ubiquitous in non-human primates, as well as cats and cows. And I don't know what the, how prevalent they are in horses, but there is a horse Foamivirus. So it's kind of interesting that all the all the Foamivirus species also have or the types of animals have lentiviruses. But Foamiviruses are present in all non-human primates, and they've been dated back. I don't want to get into a controversial discussion about evolution of primates, but guessing 50 to 60 million years. They're very ancient viruses. And as far as we know, in their natural hosts, they're totally non-pathogenic. They don't even cause symptoms. It's hard to know if they cause symptoms, since pretty much every non-human primate in the world has Foamivirus in it. So there's not much to compare it to. In order not to be infected as a non-human primate, you would have to be kept alone for your whole life, and then you can't really be compared to social living animals. So they've co-evolved with their hosts for 50 to 60 million years. They're extremely successful. As I said, all NHPs, all cats, all cows are infected. And they don't even have to cause any symptoms. They are secreted into saliva, and all of these animals share saliva, either from bites and licks in primates and cats, or through sharing cud in cows. So they're extremely successful, because if you want to be a good virus, all you want to do is transmit as well as you can. And if you don't make your host sick, this is better. They'll be living longer to transmit you to all the young. And if you look at primates, and this is also true in cats and cows, all adults are infected. We know from our primate studies, and it's also been shown for the other species by other people, that juveniles and young adults are not infected, and that's because they get maternal antibody from their, well, obviously from their mothers, and this prevents infection. So amongst macaques that we look at in Asia, by the age of 3, all the animals are infected. But if you look at animals younger than 2, they're mostly non-infected, and they start getting infected between 2 and 3 years of old. Okay, so we had gotten the virus from Flugel, and I brought it, and his sequence was out, and I had it at the Hutch, and I was talking to Mike Emmerman, and he said, oh, it has a transcriptional transactivator. You can make a cell line to assay it. And until then, it was this really tedious endpoint dilution and looking for cytopathic effects. I must say that in vitro, almost every cell line is killed by foamy-virus. It's highly cytopathic. It's hard to get latently infected cells. But this is not a good system because in vivo, almost the cells are either latently infected, and it only has a very small niche of replication, which I'll get to. So what you learn about foamy-virus in vitro, the biology, doesn't reflect what's really happening in the real organism. But anyway, he had just come up with MAGI cells, which I think is multinuclear activation of galactosidase indicator, and he gave us the beta-gal with the NLS in it, and we hooked up the PFV-LTR and put it into BHK cells, and we had a nice indicator cell line, which made all our studies really easy, much easier to assay virus and look at the effect of whatever we were doing to them. I must say there are no vertebrate cells that cannot be infected by foamy-virus that I know of. Okay. Okay, so that was our first paper on foamy-virus was 1990, either 3 or 4, I'm sorry, I can't remember, maybe about 1993. And in 1994, I asked Robin Weiss if he would, he was working with Myra McClure a bit on foamy-virus. I said, could you please have a foamy-virus meeting, because nothing makes sense. When you look in the literature, there are no northern blots to show there's RNA. There's no GAG-Pol fusion protein, and GAG is never cleaved to smaller matrix capsid, nucleocapsid. Can you please, I want to talk to the people. So he convened a meeting in 1994 in the fall outside of London with Myra. I get to the meeting, and we're going to present on our new cell line, and they're all the people, mostly Germans, a few English, French, but very few people from the US. And I said to myself, okay, I've been in this field a long time now, like over 20 years. And I got up, they're just going through all this stuff, and I said, okay, where's the RNA in the virus? They said, oh, we just don't have good enough techniques yet. It's coming. Where's the GAG-Pol fusion protein? Oh, our antibodies aren't good. We know there'll be a GAG-Pol fusion protein. Why isn't GAG cleaved? Oh, we just don't have good antibodies. Everything. And I just went back to the lab with my postdoc, Allison, then the Shuyarn Yu, and I said, there's something wrong here. We've done thousands of northern blots. We can't find RNA. We've done thousands of western blots and IPs. We can't find GAG-Pol or fused GAG proteins. And so we sat down and looked at the sequence. Well, it turns out, the GAG-Pol of foamy-virus, here's GAG, and Pol is in a +1 reading frame relative to GAG. Well, I went through the literature, and there was no frame shifting in the +1. It was all -1, or as you know, some of them have the suppression of termination. So we looked more carefully, and we said, ooh, it looks like there's a 5' splice site in GAG and a 3' splice site right at the end of GAG, so we could get a Pol splice message. And we made mutations in the 3' splice site, and the virus was dead. There was no Pol. So we showed that Pol is made from its own splice message. And we then took the virus, and we were just, really thinking about how in 1982 or approximately, Mason, the same person I'd worked with in the Vogt Lab, and Jesse Summers, had shown that hepatitis B virus was really like a retrovirus. It packaged RNA, it had reverse transcriptase, and everyone thought it was a DNA virus. I must say that it's my understanding when that work was submitted to this meeting, it was given a poster. Anyway, so we thought, okay, let's do southern blots. And we did southern blots on supernatant pelleted virus. And there was a 12 kb double-stranded DNA. And later, in 1996, we used AZT to show that unlike HIV, the only reverse transcription that occurred that was important was in the producer cell, not in the infected cell. So foamy-virus is really a DNA virus, although it packages RNA, it reverse transcribed while it's budding from the cell. That was really exciting, and I could see that the lab was just going more and more into foamy viruses because for several reasons. One is they're present in so many species of non-human primates. What about zoonosis? If you look at the lentivirus story, the natural lentiviruses are not highly pathogenic. Maybe there would be a recombinant and selection in humans. So we got really interested in zoonosis. And these viruses have co-evolved with their natural host for 50 or 60 million years, and they've done it in a way different from other retros or hepatinoviruses. And people have gotten really interested in foamy virus vectors because they have large genomes, they're non-pathogenic, and they're actually DNA viruses. Maybe they would be better vectors than RNA viruses. So we decided to really do a lot on the molecular biology and also the in vivo biology of foamy viruses. So we made some other very interesting observations that make foamy viruses pretty unique, and let's see. I'm just going to say,
that's a good spot.
I just want to point out the people in my lab currently. Allison, unfortunately, has left the lab. I will tell you about her most recent work. Eng-Young Lee, a staff scientist. Xiaojing Wang, a postdoctoral fellow. Who's next? I can't see. That Carolyn Stenbach? Oh.
So
Carolyn Stenbach, a former graduate student who is in Seattle U and is doing a sabbatical in the lab. 3 technicians, Dana Jackson, Kane Sullivan, Sergey Ivanov, and my collaborators at University of Washington who are so important for the zoonosis and phylogenetics, Lisa Jones-Engel and Gregory Engel. And I'm going to tell one more little story about me. When we started, Lisa is a primatologist, Gregory is a physician. They were going out into the field and getting samples. And I would say to them, oh, you guys just go get the samples and we do all the hard work in the lab. All the Western blots, ELISAs, PCR, until I went on a trip with them to China. And we were just in a wild animal park. We weren't out in the woods. And it was the amount of work that goes into getting monkey blood and buccal swabs is unbelievable. And so when I came back, I said, oh, they're doing all the hard work in the field, and all we do is a stupid PCR. Okay, all right. So, so I mentioned that so the current work in my lab, and I'm not going to talk too much about the phylogenetics and zoonosis. We have 2 projects, an R01 that Lisa's PI on. We're looking in so I should back up. In the early 2000s, we started looking more at the biology of foamy virus. At least in vitro, in cell lines, we show that the BET gene, this other non-structural gene along with the transactivator, we found that it was required for induction of virus from a latent state. There are a lot of papers in the literature suggesting that BET might be an ApoBec inhibitor, but I think the data is on both sides, and if I had to guess, I would think it's not have anything to do with ApoBec. We also had a graduate student in my lab, Shannon Murray, who repeated some work done in Germany, looking at the tissues these were macaques from the Oregon Primate Center where the virus is latent and where it replicates. And she looked very carefully and found that in the blood, white blood cells, about 1 in every 10 to the 4th to 10 to the 5th cells has an integrated copy of foamy-virus, but there's absolutely no evidence for viral replication anywhere in the animal except in the oral mucosa. Okay, and this is how virus is transmitted through saliva. And she further showed that the only cells in the oral mucosa that produce foamy-virus, and in vitro it's highly lytic, were the superficial epithelial cells of the oral mucosa that were already differentiated, cells that were probably destined to die and sloughed in the saliva very soon anyway. And so these sloughed cells and virus in the saliva is how the virus is transmitted. And she found no evidence for RNA anywhere else in any of the macaques, except in SIV 239 totally immune suppressed macaques that they were doing HIV type studies on. When we looked at the necropsy tissues of these monkeys, we found foamy-virus replicates in the jejunum, a major site of T cell depletion. And don't ask me anything more about it because people in the field don't want to even acknowledge that foamy-virus could have anything to do with SIV pathogenicity in macaques. It's like a little secret that they won't talk about. People report that there are syncytia in the jejunum after SIV pseudomongoby infection, and it could be that foamy-virus. They often die of intestinal disorders. So foamy-virus could be a cofactor, but I don't really know. I'm just throwing that out. So, and then Lisa and I did a bunch of studies with Gregory and others looking at transmission of the virus in Asia and also zoonotic transmission. And I just want to say that our current studies are focused in Bangladesh, and I'm just going to give you 2 facts. And there's so much data that I don't understand. I'd be happy to talk to people about it. It's giving me headaches at night. But if you take Bangladesh, and we've looked at 5 or 6 different villages or regions. So in Bangladesh, people do not bushmeat hunt. They're mostly, there are a lot of Hindus and Buddhists who, would not, the Hindus really love monkeys. Hanuman is one of their main gods, the monkey god. Buddhists, neither. There's no bushmeat hunting. These people just build their houses out in the country where the monkeys live. And so the interactions are completely casual and non-induced by people, except they live there. And we find that if you gave us a monkey from Bangladesh and we sequenced the foamy-virus from the peripheral blood, we could tell you where that monkey came from. It is as good as microsatellite RNA to place an animal in the geography of Bangladesh. And we found that there's one region in Bangladesh where about 8% of the humans who report interactions with monkeys are infected with foamy-virus, as opposed to all the other regions where it's 1 to 2%. And so far, we seem to see that the virus in this region seems to be in a higher concentration in the saliva, and it's also somewhat different. And we're really interested in why why people in one region get more foamy-virus than in other regions. So this 8% is equivalent to bushmeat hunters in Africa who are, they're mucking around with monkeys in a serious way. And The other thing I want to talk about, and what goes around comes around, how much time? Oh, I better hurry up. Okay, so in 1973, my first paper was on TS mutants of RT, and my very last paper from my lab in 2012, just being published now, is showing what happens when you make foamy-virus into a GAG-Pol virus. So we deleted one nucleotide between GAG and Pol and put them in the same reading frame. And I must say, Dirk Lindemann in Würzburg, wherever, was publishing at about the same time similar studies. We found that if you make a GAG-Pol foamy virus and you co-express it with GAG, so that it has GAG and it has GAG-Pol, it has reasonable infectivity within a log of making a splice Pol. So we know foamy viruses can use GAG-Pol. And one of the main questions we're interested in now is, if you make a GAG-Pol virus, when does reverse transcriptase occur? Does it occur during assembly like a foamy virus, or after infection like other GAG-Pol retroviruses? I don't have the answer yet. We're doing these AZT experiments now. Another interesting question is how this Pol gets packaged. Pol, there's, as Axel Rethwell mapped a Pol packaging signal in the RNA of foamy viruses. It's very close to the cis acting signal, the Psi signal, at both the 5' end of the genome and in the Pol gene. It's a complex structure, and this sequence is required for Pol encapsulation. And what my lab showed was that the first GR box in GAG is absolutely required for Pol encapsulation. So we think maybe a Pol-GAG complex binds to RNA. This packages the Pol. So there are many few Pols in normal wild-type infection than in another retrovirus. And in collaboration with Steve Hughes and Paul Boyer, we showed that foamy virus Pol is much more active and processive, probably because there are only a few molecules. If a Pol dimer binds to each RNA, that would only be 4 Pols per virion, as opposed to hundreds in other cases. And we don't know in the GAG-Pol if it's going in that way or through GAG domains co-assembling, and that's another thing we want to look at. And then the last thing I want to talk about is, so in 1978, I never thought I'd be working, well, we've done a lot on Pol, but it's sort of the ends of the 5' and 3' ends of my career so far are RT. And I also want to say in 1978, Psi, I thought, and we showed along with other groups that, mostly Dirk Lindemann's lab, that 2 GR boxes are required for packaging. And I said Axel Rethwell mapped Psi in the RNA. So I thought, that's the end of packaging, okay? But we were very intrigued by work of Suzanne Standmeyer and others working on yeast TY, that these viruses, that P-body protein seem important for viral assembly. And we had shown that foamy viruses they assemble intracellularly. I hope I can do this. Is this a nucleus in the cell? Okay, and I'm putting a little spot here, and I'll tell you what's in that spot. We showed that they assemble intracellularly, and they have a CTRS sequence like is found in beta retroviruses, at first, I think, defined by Eric Hunter. And we find that the GAG and RNA are in that little spot. And that spot co-localizes with, I'm telling you, the people doing the IF are better than me. Co-localizes with gamma tubulin. So we originally called the SGA the site of gag assembly, but now we think it's at the Microtubule Organizing Center, or MTOC. And Paul is also there. So that's where the virus assembles. And because TY also assembles intracellularly, we thought maybe some of these Peabody proteins would be important. So I contacted Vineet, who had just come up with, or had siRNAs to a bunch of Peabody proteins in human cells, and he kindly sent us 6 of these siRNAs, and we tried to knock out 6 different Peabody genes, and we found that 3 of them had an effect. I'm only going to talk about one of them, and this is DDX6, which is a dead box helicase. And if we knock out DDX6, then we get wild-type levels of GAG and RNA in the cell. Particles they're about normal levels of GAG-containing particles that can be precipitated from the supernatant, but they're devoid of genomic RNA. So we think DDX6 is required for RNA packaging in foamy viruses. Interestingly, if you look at infected cells, about 20% of the DDX6 is no longer in little granules in the cytoplasm, but co-localizes with GAG, POL, and RNA at the MTOC. And we said, oh, this will be really nice. Maybe it's a transport to get one of the like the RNA to the MTOC. Well, when you knock out DDX6, you still get GAG, POL, and RNA at the MTOC. So it doesn't have anything to do with transport. And our model now is that DDX6 has something to do with RNA or GAG RNA structure that makes it available to start assembly and packaging. This is what I'm hoping, if I get my grant funded, to work on. And we did use a DQAD mutant. If you knock out DDX6 endogenous, you can express exogenous DDX6 and rescue RNA packaging. If you do DQAD, it doesn't work. So RNase H ATPase active site is required for foamy virus RNA packaging. So again, early in my career, 1978, started working on packaging, and now here we are. What goes around comes around. We're working on packaging again from a different point of view. I know that DDX6 does not just be a packaging factor primarily in HIV, but for foamy virus, it looks like it is. So I think that's, yeah, I think I talked a little too long or something, and I really want to thank the organizers for inviting me, even though this has been, I've been up nights worrying about this talk, and you know, I can't sleep well anyway because being blind, I'm depressed all the time, and then just the thought of getting up here and doing something stupid, so. But now it's over. Where's my pole? I stick. Shall I take questions? Okay. Okay.
You can stay here. You can stay here. Questions for Maxine?
Hi, Maxine. This is Welkin Johnson from Boston College. I had a question about the studies in Bangladesh, whether or not you can tell, is there any evidence that the virus is spreading human to human at all? Or is it all dead-end transmissions from the monkeys?
That's a very interesting question. We're very interested in this question. It's very difficult to do with, you know, the people are in Bangladesh. I'll tell you about this new R21 in a minute. I will say that CDC and a few of the people working in, is it Gambia or somewhere in Africa, have looked for DNA in the oral mucosa of infected humans and could not find any. We have looked in the buccal swabs of 2 humans that we've had trouble. As I said, my collaborators are a primatologist and a physician epidemiologist, and they brought back the RNAs in the wrong buffer, so we've had a little trouble. We're getting new samples. We've looked in 2 humans and we don't find any RNA, but then in some monkeys we don't either. So the new R21 this is right before I got squashed by the car. My collaborators had been in the American Society for Primatology meeting, and they brought back blood and buccal swabs from a number of American primatologists. And I looked, and it's a long story, but tentatively I found that some of these primatologists are infected with both Old World NHP and New World monkey foamy-virus. We thought, this is incredible. The only place these 2 kinds of monkeys will come together in labs or primate or zoos, and if these people are multiply infected, we want to look at them for recombinant virus. And Lisa knows the names of they signed informed consent, and so these are the population we're going to do more of that study in to see if they have any virus in their saliva. CDC could not find viral infection in the families of the 3 male primatologists they have studied who are foamy-virus infected. And one other thing I wanted to say is most people in the literature who are either veterinarians or bushmeat hunters who are infected, I think they're completely men. In Bangladesh, the people reporting bites, scratches, saliva, whatever from monkeys are 50% female, 50% male. Of the, I think it's something like 19 infected people we found, either 18 or 19 are female. We don't understand that. But a lot of the American primate handlers who might be infected, a couple of them are pregnant, and they really want us to assay their kids too.
More questions?
But we don't know. And one other thing I wanted to say is, I'm surprised nobody asked me. Every time I give a talk, they say, "Ooh, do monkeys kiss?" I won't give you the answer I normally give. But I'm just going to say we don't think it's oral. We think it's oral saliva getting into the blood through the skin. That's the transmission. Yes?
Hi, Maxine. It's Nick Meyerson from the University of Texas.
Okay.
You mentioned that these foamy viruses can infect many vertebrate cell lines.
Yes.
I'm wondering if you can comment on why you think it's such a promiscuous virus.
Well, I think I read a paper just before I came. Someone correct me if I'm wrong. They think the receptor is heparin sulfate. And there's some viruses, there's some cells like 293 T cells, which the LTR doesn't work in. But if you promote it with CMV instead, it replicates fine. So we don't know. We think it can get into every cell, and most cells tasks can be functional in. That's all I can say.
Maxine, given the high viral burden of foamy viruses and their hosts, is it assumed or presumed that some of the accessory proteins have functions that prevent recognition of the virus by immune responses or enable evasion of immune responses, and such as the one you think that doesn't interact with APOBEC? But
I tried to get a grant to infect monkeys with Bet minus virus,
Great.
but it wasn't funded. We have some this is the evidence that's making me pull my hair out, but there's some evidence that there might be a lot of G to 8 transitions in humans in Bangladesh, but not in monkeys, which makes no sense. But we don't know if APOBEC is somehow involved. But yeah, I don't know what else.
Species-specific differences could enable that sort of mutation.
Yeah.
Yeah.
And nobody's ever infected an animal purposely with foamy virus. So it's all natural, even in the primate centers. Because unless you take a young from all other adults and raise it completely alone, it'll get infected. But the point is, maternal antibody protects, but later on, it's no longer protective after a few years. And that's true in all 3 well-studied types of animals.
Okay, well, thank you for just a beautiful seminar. Thank you for making the trip out here to Cold Spring Harbor, and we look forward to chatting with you
Thank
until
you for
Thursday.
inviting me.